Great. So one of the major goals in my lab is to understand disease mechanisms and improve health through precise measurements on human bodies, and not just on the human cells in the body, but also on the microbial cells. It live in our gut, on our skin, in our respiratory tracts, and throughout many of the organs of our bodies. And there's mounting evidence that including microbes in precision medicine can do important things like better predict what doses of drugs we should take and whether those drugs would have adverse side effects. And there are many companies here in the Bay Area and worldwide that are trying to use the microbiome in therapeutic development. This is a very large scale omics problem. The precise measurements of the microbiome are usually done by sequencing, often DNA, sometimes RNA, sometimes metabolites or proteins. But let's just talk about DNA. If we take a sample, we get millions of short sequences. Often it's 150 base pair paired end sequencing. And in here, there's going to be a mixture of hundreds of species. To figure out what's there, these short sequencing reads get compared to large databases, currently with hundreds of thousands of genomes in them, each on average, say 5 million base pairs long. And we and many others write code that's going to search for matches, basically. This is hard for a number of reasons. One of them is that when this field got started, for the human gut microbiome, 50% or less of the species weren't present in the databases. So anything that's not in the database is invisible. Through some efforts I'll briefly describe, that's down much lower now. The other problem is that this search is very intense. This is much more computation than, say, doing an RNA-seq experiment on a human sample. We're lucky enough to have access to some cloud computing through the Chan Zuckerberg BioHub. We're going along doing our work and got the attention, believe it or not, of Mark Zuckerberg, who said my computer bill was too big and that he didn't want to pay it anymore. And so we were doing something about like around $10,000 a month of computing at that time. That is not fair. It's not fair, perhaps, that Mark Zuckerberg has to pay my bill, but more importantly, it's not fair because it's not democratic. It means only someone like me who has a lot of resources can do these analyses. Why am I blowing that amount of money anyway? And even if it was useful to science that I was blowing that amount of money, it means that most of the world can't do these analyses. So I'm going to show you that we got some of these analyses to run on a laptop computer. So solving the computational problem. The third problem, which is the main thing I want to talk about today, is that the analyses are very biased and give very misleading results. In particular, when there are closely related species in the database, they completely confound all the short read aligners like bowtie, BWA, minimap. And those incorrect alignments lead to biased downstream statistics. I do not have a magic bullet for this one. It's something we're still working on, and I welcome your input, and I hope others will be interested in thinking about it with me. But I will show you some mitigation strategies that we've developed. So the talk is mostly about alerting anyone who's doing this kind of work, and also anyone who's not has feedback about it to this problem, but I will also talk about some of the other stuff. Now, most people who've heard about microbiome science, and maybe some of you have done some, are probably thinking about just figuring out which microbes are there and how much of each one. That is what most microbiome papers do. It's my opinion that that is not particularly useful for precision medicine because two strains of the same species, something that would be put in the same chunk of this pie chart, can have vastly different functional capabilities. They can one can be pathogenic and one not. One of them can have create an anti-inflammatory metabolite, one might not. One of them can inactivate your cancer drug and the other one might not. And we're all kind of aware of that in this world today of COVID variants, that like every time there's new variants, they're all COVID still, they're all still SARS-CoV-2, but the function in terms of things like important things like transmissibility and pathogenicity is evolving over time. So we tend to not do these pie charts. What we do is something much more precise, but also much more computationally intensive, which is to analyze the samples at a strain level by calling genetic variants in the short reads. And doing this is what alerted me to this problem with closely related species in the database. So I'll tell you a little bit about it, because it's the context for our work. But I think the problem I'm going to share with you is much broader than this genotyping problem. So what do I mean by genetic variance in the short reads? For one species in this test tube, one place in its genome, within that stool sample, you could have a mixture of strains. So maybe 70% of the reads here carry one variant, and then there's this G that's present in 30% of the reads. So that's within sample genetic variation. There can be between sample genetic variation, either from the same community, like sampled from the same person, but 6 months later, or just two completely different people or two completely different environments. And I should say, I work on the human microbiome, primarily on stool samples, but what I'm saying applies to any kind of environmental sequencing studies. So if you're studying soil or water in the ocean, the same would apply. So you can do this genotyping. To find the short reads, we take metagenomic sequencing, we align it with tools like Bowtie, which is what I'm going to talk about today, but it's none of this is specific to Bowtie. We align it to a reference database. And instead of saying how many reads do we have for each species and making a pie chart, we just make a pile up and we call variants. Like anyone who's familiar with running like a genotyping pipeline for humans, it's the same idea. We do this iteratively for each species in the sample and each position in its genome, and we get these variants. You can also look at the pileup of reads and compare coverage to the coverage of universal single copy genes and estimate the copy number variants for coding genes as well. But I'm going to focus on single nucleotide variants. Okay, and I'm not going to talk at all about this. The fun part is once you have these, you can do some really cool things like doing GWAS, but now with the microbial variants to host traits. You can study the evolution of the microbiome. You can track strains and ask whether strains present in a baby were actually in their mother. Did the baby get it from somewhere else? All kinds of fun stuff. But I'm not going to talk about that today. Happy to discuss later. Okay, so first I'm going to talk about something I'm calling challenge zero, because this is sort of like an old problem that's been solved for a few years now, but just as some background, because this was kind of an important thing. A big problem a few years ago was that many species are invisible to this process because they're not in the database. We developed a method using universal single copy genes that should be present in every cellular organism to calculate how much of the diversity in a sample is represented in the database. So if you were to do read alignment to a database, how much would you see of what was actually there, what proportion? And so you want that to be 100%. And you can see for samples from a whole bunch of natural environments, it's really abysmal. It's better for the human microbiomes. Some environments like skin, we know almost all the microbes that are there because it's a simpler community and it's been well characterized. Human stool has this huge variance. You can see the bar there. And what we showed back in 2016 was just like in human genetics, where there's been a bias to study Europeans and North Americans, exact same thing in microbiome research. The database is biased towards North Americans and Europeans. So these samples from Tanzania and Peru, for example, had very low database coverage compared to the ones from US, Europe, and also China has sort of joined the game in this field, at least, as being like in the database instead of out of the database. But basically the rest of the world, besides the US, Europe, and China is really poorly, was really poorly represented in the database. This launched a huge effort to try to get better coverage, better representation in the database. And while isolation and culturing in the lab is still very biased to certain geographic regions, the shotgun metagenomic sequencing, where you just get stool samples and sequence everything in it, has been less biased. I wouldn't say unbiased at all. You can see the reddest places on the graph are still China, a few European countries, and the US. But there has been sequencing in many countries on 6 continents. And so we and others joined in this international consortium, assembled genomes from these shotgun samples, and we've been closing the gap. So this is the amount of gap closing that's happened. And the really rewarding thing is there's been a lot more gap closing for the exact places that were poorly represented in the past. So it's there's still bias, but it has been greatly improved in the last few years. And through efforts like this, as well as single cell sequencing and something called culturomics, where people try to grow bugs in the lab that they haven't been able to grow ever before with some more advanced microfluidics, there are now genomes for a lot more species than there were a few years ago. There's been an explosion since 2015. This is NCBI assembly, which is not including the metagenome assembled genomes. And then here are 3 databases that the UHGG, the GTDB, and the GEM are ones that use these metagenomic samples. And the important thing for this talk is there are now many, many species that have hundreds or even thousands of genome strains that have a genome sequence. So there's a lot of interest-specific diversity in the database. Okay, so more genomes sounds like good news. Is it good news? Well, for the problem that I initially started with, yes, it means now for a human gut microbiome, even one outside of North America and Europe, the alignment rate is much higher. So there's less, it's not solved, but there's less of these invisible species and there's less of this geographic bias. However, 2 new problems arise. So I'm going to talk about both of those. The first one, and the main thing I want to present today, this is sort of my main point, is that we now have a densely sequenced tree of life where many species have a close relative. And what happens when you align short reads to such a database is the species are competing for reads. That will not be news to anyone who's worked on like RNA-seq and knows about multi-mapping. This is sort of like multi-mapping on steroids because there's multi-mapping within every species and then also across species. So this is quantifying the amount of closely related species, the sort of greener or bluer, as we move from yellow to blue on the spectrum, there are more species in each lineage that have a neighbor that is within 92 to 95% average nucleotide genome wide. 95% is sort of a rough species boundary or one that's operationally used. So these are things that are just past the species boundary. So certain lineages have a lot more than others. But the point is that there are thousands and thousands of pairs of closely related species now. Here's what happens in theory, and then I'll show you the numbers on real data when you have a close relative in the database. So the first thing, which is probably the most cryptic one, is the read. So dark purple is like the correct or on target genome. The light purple or fuchsia is the off target, the closely related species. There's the purple read getting aligned, the red dots are the SNPs. The percent identity is shown, and then the outcome is sort of described below. So in this first example, it's aligned. In fact, it's aligned to the correct species. But the statistics that the aligner calculates in bowtie, that would be the map ID or the score, and the map Q, which sounds like quality. So we all use it for filtering. The quality of this alignment is a sort of medium to low because the next best alignment, the second best, is almost as good as the first. So map Q is the ratio of the best alignment score to the next best alignment score. And so if we were to quality filter, even though this is correctly aligned, it would likely get filtered out. These 2 are worse. So the read won't be aligned at all because there's very close competition or even perfect, exact match between the best and the second best alignment. And then here's the thing that seems like it shouldn't happen, but it does, is the read can actually map to the wrong species with a higher alignment score than the correct one. This is called cross mapping. Okay, those are all bad. And here is how much they happen. This is a simulation, 300 species. In each case, the closest neighbor starts very far away. And as you move this direction across the plot, it gets closer and closer to the species boundary. So your neighbor in the database is getting closer and closer. And then the color is how diverse is your species itself, meaning the strain in the reads, when it's red, is pretty far away from the strain in the database. Whereas here, they're the exact same strain. And so you can see 2 patterns. One is close neighbors are really stealing reads from the on-target genome. You can have as much as half of the reads going to the neighbor instead of to the correct genome. And that happens more often as they get closer, which makes sense, and more often when the strain in the sample is diverged from the thing in the database. So the Tanzanian sample being aligned to the European reference database, for example. So that's a quantification of how much this happens at the alignment level. Yeah, Gian.
So this plot is going to change the read length.
Yeah, so these are 150 base pair paired end reads. And right now I'm treating them as individual 150 base pair reads. In a minute, I'm going to say that using the paired end on the next slide, that using the pair can help a little bit. And in my conclusion, I'm going to say long reads will help a lot. So yeah, absolutely. There's hardly any long read sequencing in this field right now. It's almost all 150 base pair paired end. So that's what we simulated. Yeah. So great point. Okay, so that's what happens at the alignment level. Here's for us when we use these reads to call genotypes. I'm not showing you precision. I'm just showing you recall here. But when alleles differ from the reference, so you have an alternative instead of a reference allele, you have a really big degradation of recall, your ability to call those alternative alleles. And that gets worse as it gets more purple, which means as your neighbor is getting closer. So as your neighbor gets closer, and as you're calling alleles that are not the same as the sequence in the reference database, we have a pretty low recall. There's the same pattern, but not as bad for reference alleles. There's also bad precision, which I'm not showing you, and it's because of the cross mapping. So if we want to reduce the cross mapping, we can put a MapQ filter on. We found a MapQ of 10 reduces the cross mapping and improves the precision, but it makes the recall even worse. So the first one was like no MapQ filter at all. This is with a MapQ of 10. If you say that one of the reads in a read pair has to have a MapQ above 10, but the other one can be even worse, then you can maintain your precision, which I'm not showing you, and you get a bit of your recall back. This is not great though. Like these recall, you're missing a lot of SNPs. So this is what I would call a mitigation. Like this is a recommended thing to do paired end filtering and the MapQ of 10. It's not a great solution. It's just the best that we could come up with. Essentially, this is like a rock in a hard place problem. But the point here is that the alternative alleles suffer way more than the reference ones. The other thing you can do is to try to get the species in the database that are close relatives out of there if you don't think they're in the sample. So if you have a close neighbor and you don't think that's in the sample, but you believe that the organism you're interested in is in the sample, get the close neighbor out of there. That makes the problem a little bit better. So it doesn't help a ton because sometimes the close neighbor is in the sample, but it can help a little bit. So those are our two mitigation strategies. This is not great news, but this is something we recently published that is an improvement over what everyone has been doing. Okay, so the mitigation strategies help, but basically we need to avoid doing alignment with bow tie. So I'm going to show you another solution we came up with for our specific problem of genotyping. It may work for some of the other problems, and it avoids doing short read alignment. The idea is to develop a database of short probes or k-mers that you're going to use instead of alignment to entire reference genomes. And you want to pick those k-mers so that they are unique, that they do not occur in other genomes. What we're giving up on here is that we are now not going to align reads to the entire genome. We're going to align them to like the equivalent of like a genotyping array, but on the computer, rather than say whole genome sequencing and then calling SNPs in the pileup. This is like a computational genotyping array, or even like a microarray with probes on it for people who are familiar with that. You want your probes to be unique so you don't have cross hybridization, but you're giving up on like the quantification and the genome wideness of doing RNA-seq. Okay, so we design these probes by doing genome comparisons, and we put those probes through a filter where we're trying to find out if they're really unique and occur nowhere else in the database. It doesn't mean they don't occur anywhere else in nature. We cannot guarantee that, but we guarantee that they've never been seen to the world before and have been like all of sequencing that's ever been done, that they only occur in that place, in that genome for that species. There are some single nucleotide variants that have no unique k-mers. So we also have an option for imputing with LD that I'm not going to talk about. We also, at the same time, tried to solve the computational problem. So this solution is much, not only, as I'll show you, more accurate, but also faster than alignment. We do that with two things. One is that the k-mer exact matching is a very efficient algorithm. It uses a prefix filter that's sort of like a Bloom filter, where we look at the first few base pairs and then only. Then we have a collex ordered and a sort, where we can go into this array at a specific place and not search through the entire thing linearly. And then we work with an exact match based on suffix arrays. So this is a very efficient algorithm. We also have an efficient data structure where we don't store all the k-mers. We store a span around each SNP of where any k-mer could occur, and then an index into that sequence. And we compute on the index and then only pull out the actual sequences when we need them. Okay, all of those things made it about 6 times more compressed than bzip2, and as I'll show you, much faster than alignment.
Okay, are you using?
So you can try different lengths of K for this application. Across all bacteria, we found 31 was a good solution. But the K is actually, so this program, the software dynamically chooses K for you based on your hardware, because it also depends on your hardware. So in terms of like this trade-off of storage versus compute. So if you have a lot of RAM, it will let you use a bigger K, basically. Okay. Oh, that last point was, I'm talking about short reads like a metagenomic sample, but this exact same strategy can be used for comparing genomes to each other, assemblies, contigs, unassembled reads. Okay, it's about 2 orders of magnitude faster than alignment. Our method is in green. The other methods are all alignment based, except for I'm showing here Kraken-2. Kraken-2 doesn't do genotyping. It does just like taxonomic composition, and it's also k-mer based and was part of our inspiration for this. And so I'm showing it there just to show another k-mer based method that solves a different problem. So we're much faster. We do use more RAM. And the reason that this runs on a laptop is that we're going to store all of this in the computer RAM. We're going to operate on it there. And there's not a lot of like reading and writing to disk. And so we use a fair amount of RAM, but we are very careful to choose this number. Oh gosh, it goes off the screen, but it's, well, you can see here for about 40 gigs, or for us, about 25 or 30 gigs of RAM. So you can run it on a kind of beefy laptop. It is also more accurate than the alignment-based methods. So in gray and brown are 2 alignment-based methods. In green is our method. Has lower false discovery rate and comparable power or sensitivity to the other methods. Because it's fast, we were able in a couple of weeks for a much smaller AWS bill to be able to analyze 7,500 samples. This is just an example of a great deal of data went into this. So each of those samples is like 50 million reads. So we're going to analyze hundreds of species. For every sample, we're going to get a genotype for every species, stack up those vectors, and then say how similarly related are individuals to each other based on their microbiomes. This is a genetic relationship of the world population based on their microbiomes, not on their human DNA, either as a heat map or here as a UMAP. Okay, so unique k-mers beat alignment at known SNVs. The last point I want to make, though, is that this method that I just talked about, which is kind of neat, depends on having this like pre-computed database of SNVs. It only works for known SNVs. So now, once again, anything not in the database is going to be invisible to this tool. And that is definitely a problem. So it also means that we have to do this whole genome multiple sequence alignment, or at least pairwise alignments, to discover the SNVs. So that's very computationally intensive. How can we align and call variants in so many genomes if we have like 300,000 genomes? Tandy talked about the difficulty of alignment today, for example. Okay, so very quickly, what we did was develop an algorithm to pick a subset of all the genomes. So just think about one species now. Let's say it has 1,000 genomes in the database. I want to pick a subset. The optimization that we're trying to solve here is a minimal subset of those genomes that captures as much of the common genetic variation as possible. So what that means intuitively is you want to find genomes that are not related to each other, that are sort of spread out across the space of genetic diversity of that species. So to do that, we're going to cluster all the genomes, and then for each cluster, pick one representative. And I'm not going to have time today to talk about the algorithm, but this is based on doing a fast clustering of the genomes with something called Maast distance, which is an approximation of average nucleotide identity, and then trimming the graph using this algorithm define something called a D-cut, which is basically like where I'm going to cut edges out of the graph. Once I have those representative genomes, one for each species, I can see how much redundancy was in the database. So here, this species A. putridinus had 3,646 genomes, but I only need 46 clusters. And I'll show you in a moment that that captures most of the genetic variation for this species. In contrast, A. rectalis over here didn't compress as much. A little over 5,000 genomes, I still needed 2,650 clusters. So this is a data adaptive choice of how much to compress or how much redundancy to remove from the database, with the goal of still being able to capture as many SNPs as possible. So here's the genome, the species with 46 clusters. SNP discovery maxes out pretty quickly. So if we go to 46, we're getting most of the genetic variation. There's the species with more, and it takes longer to max out to the diversity of that species. And so you're going to need at least a few hundred clusters, if not more, to capture all the genetic diversity there. I don't think I have time to talk about this in any detail, but we also showed that using these representative or tag genomes actually decreases bias. If you want to discover all SNPs below a certain minor allele frequency, like 1% in the population, but your database is really biased, you have like 300 genomes that are all from the same hospital outbreak, they're all very closely related to each other, then you're going to misestimate the minor allele frequency. You're going to think anything that's in those genomes is more common broadly in the population than it really is. And so what people commonly do is pick a MAF threshold. And what we showed, and I won't go over the graph here, but we showed that we actually have a less biased estimate of the true set of SNPs at a given MAF frequency. Finally, this is faster than other approaches for calling SNPs in genomes. These two are the competing methods. These are two versions of our method. The competing methods are slower. So this is elapsed time. They're slower for 500 or 1,000 genomes, and they fail on 5,000 genomes, whereas ours runs. And then you can run GTPRO or other methods to genotype those SNPs in genomes very fast. So we can genotype very quickly, like a large number of SARS-CoV-2 genomes, or what I'm showing here is a little over 3,000 H. pylori strains, a bacterium that has really interesting biogeography across the world, and build a tree like this. Cool. Okay, so tag genomes speed up variant discovery. They also improve accuracy. So it's great that we've been all doing so much genome sequencing and assembly, but you don't really need the next genome that's just as similar to a genome you've seen before. We really need to focus on new lineages and not redundant ones, because they don't have a lot of value. And in some cases can really confound your analysis. Okay, so I just wanted to stop and say, like, we're happy with the tools that we built. They've just been released. You know, they help this situation, but we have not solved this problem of short read aligners failing on big data sets with big reference genome databases. So just stepping back a little, like from our own work, I just wanted to conclude with a few thoughts for the field. We definitely need some different strategies. There are some that are already out there that many of you know about or even worked on, like genome graph algorithms. They would be really useful here. Right now, they are not fast enough to work with this much data. Similarly, probabilistic read mapping, where, you know, in transcriptomics, you sort of find a multi-mapping read, and if one of the transcripts is looking from uniquely mapping reads to be very abundant compared to the other one, you have a higher probability for that multi-mapping read to go to the other transcript. The same kind of thing would work really well here. Again, those algorithms would need to be much faster to work on this amount of data. There are some neat methods coming out where there's no database at all. Julia Salzman talked about one of these in her talk at the first week of this conference. It has some preprints out. There's a few other people thinking along these lines where you just directly compare reads to each other, no reference database until you get to the interpretation stage. And then as Gian smartly gathered on like slide 2, longer reads or like haplotypes will also help with things like the cross-mapping problem. In the meantime, what we're working on and what we advise is that tools that do use things like Bowtie and BWA need to be flexibly implemented so that people can very carefully choose the thresholds that they're using. A default map Q of 30, which is what almost all the software out there uses, would be really in a bad place in all the graphs that I showed you. So I talked about bacterial communities, but other microorganisms are also starting to have very densely sequenced lineages. And as we also heard from Harris at this meeting, many eukaryotes as well. So this is not like just a microbiome problem. It's not a problem specific to the genotyping that I'm working on, but I think also just any quantification of abundance of species or of genes would suffer from these problems. And as databases get bigger and bigger, we all feel good, like, oh, more data, but it's making bioinformatics like less and less accessible to the average lab. So I think we're all obligated, people like myself who have a lot of resources, I feel obligated to think about how to make cutting edge analysis available to everyone. So here's my team. And funding, I want to especially acknowledge Chunyu and Jason, who did a lot of the work today. Thank you very much, and I'll take questions. Thanks, Lauren.
Some focus on the CO2 footprint of what we do.
Yeah.
I didn't use that argument, but I guess it's also
no, I think about that too,
important.
$10,000, but also a lot of CO2. Yeah.
There's some nice papers about what can be done in that space in bioinformatics recently. So questions here?
It's quite amazing. I didn't realize this microbiome compute is so big. For tagging the genomes, like when you have more and more genomes, can you like gradually add newly tagged genomes, or do you have to go back and revamp your previous tags?
Great question. Yeah, we have an iterative update that you can do. So you can quickly see if they belong to an existing cluster. If they don't, then you start a new cluster, basically.
Okay.
So they either don't become a tag or they do, based on their similarity to the existing. And that's a quick, relatively quick compute because you're not comparing to all genomes, just to the tags.
Interesting. Great talk, Katie.
Thanks,
And more of a biological question, I guess, for the cases where you use a lot of clusters to represent like a gene because there are a lot of SNPs. Do you tend to see clinical correlates of strain? Do you tend to see more often that those more diverse genes are more correlated with disease? Like,
really good question. So like I had that quick slide at the beginning of like the fun part, and what I really want to be working on is associating all this with phenotypes, either of the microbes or the hosts. Yes, when there's a lot of genetic diversity, there is more opportunity for strain specific associations with things like drug response and phenotypes. There's also more power. So there's two things. I think there's biological reality of like more genetic diversity, more opportunity for phenotypic differences between strains. But there's also in general, like more data, more power. So both of those come into play. So we, in our association studies, tend to focus on a much smaller set of like 30 to 50 species where we have good power and there's a lot of diversity. Just for first pass, low hanging fruit kind of thing. Yeah.
So from the practical standpoint, so how really relevant is this problem of assigning the correct rate to the correct neighbor species in terms of functional implications? Very similar species are going to have similar impacts, right? So, and for broad taxonomic characterization of the metagenomic samples, probably this is, I mean, how important is really this in the practice? I'm pretty sure that there are cases in which this is crucial, but I have not really an idea.
Thanks for the question. It's a commonly held belief that you know, maybe even out to the like phylum level, that things are functionally similar to each other. And certainly at like a genus level, like a lot of microbiome analysis is done at the genus level, and it's assumed you just need a pie chart of how much of each genus, and you can associate that with traits. There've actually not been very many replicated associations, and there are some. There are almost no mechanistic associations at, say, a genus or a family or an order level. Like no one can explain why. On the other hand, there are many examples where two strains of the same species have a different phenotype. So E. coli is in all of us, but only some of it causes food poisoning. You know, you can think of many examples. So I think I have a particular and biased point of view, but I think there is a great deal of genetic variation below the species level. We've quantified that. So for the human gut microbiome, 2 people living in the same city in, say, Europe or the US or China, we can ask what percent, if they both have the same species, what percentage of the genes do they share? That varies across bacterial species, but for some of them, it's as low as 40%. So we call them the same species, but 60% of their genes are different. And so there can, of course, then be a lot of functional differences. So on the other hand, there are some, I showed you the one with only thousands of genomes with only 46 clusters. That one's pretty clonal. So for that one, it doesn't matter so much. So it's kind of a biological question, and it depends on the bacterium. Does that answer for you?
Katie,
Yeah.
I'm first, thank you. Fantastic talk. I'm curious about, getting to ground truth and especially errors in the sort of annotation databases. So E. coli and Shigella are a good example.
Yeah.
Like, it's a mess. I mean, it's just an absolute mess. And so, as you try to go and take this into clinical implementations, we've seen, so I was part of a group that was doing like metagenomic sequencing ab initio as a way to detect infections. Turns out to be a really tough, if not ill-posed problem, right? So, and it starts with this, but then it, there's all these other issues about what do you expect, what are the priors. So I'd love to get your thoughts on how to think about that problem, particularly when, the kind of stuff that Joe does, right? Like we don't know what the infection is, and you have to have sort of some threshold in thinking about how to detect, and then you find something, is it pathogenic or not? So
totally. In a way, what you're talking about is the same problem that things, even when you get down here to like species nodes, they have close neighbors. There isn't really a species boundary. You just have a cluster of genomes and a spectrum of phenotypes across all of those. So it's sort of related to this. But my perspective on what we need to do clinically is kind of like these probes in my virtual genotyping array. I think we need biomarkers that are unique and identifiable and are clinically associated. So what those are likely to be is like gene sequences, for example, where it is the pathway, it is the enzyme that inactivates the drug. Okay, test for the enzyme. Who cares what bacterium it's in? Because it's probably getting horizontally transferred anyway. Just test for the enzyme, or even for the active site of the enzyme with some kind of a PCR probe, basically.
Which is not unlike what the bacteriologists have done for 30 years. Like, let's go look for the plasmid. If it's got a drug resistance plasmid, kind of does, doesn't matter where it came from, right? We're going to still treat it in this.
Exactly.
It's done sometimes very empirically.
Yeah, and also, I mean, a lot of not all, but many of the ways that bacteria interact with host are through secreted things like metabolites or protein or peptides. So maybe you just want to test for those rather than the bacterium. So same for therapeutics. There's a lot of startup companies in the Bay Area trying to make cocktails of specific bacteria. If we knew that it was a particular metabolic pathway that was making an anti-inflammatory compound, let's just give them metabolite, So my feeling is, I guess it's maybe a little ahead of where knowledge is, but we need to focus on those clinically actionable things rather than on taxonomy, which is sort of meaningless and very confusing.
Good, thanks a lot. Let's thank the speaker again for a great talk. And as this is the last session before I hand over to the organizers, I think we should really thank them for this meeting and also bringing the culture in our field back, the interaction, the dinners and everything. So I think it's really been a nice event. So I leave from here in a very uplifted way. So thank you to you for that. So thanks.
Thank you very much, Søren. All right, well, it's already time to conclude the workshop. I thought these 4 days actually went by really quickly, especially after such a long anticipation, more than 2 years, right? Or 3 years, I think, since the planning started. So I was actually so impressed by the quality of the talks and the discussions in and outside of the session. I thought there was remarkable breadth and impact. and the topics that are being addressed, and enough exciting questions and challenges to keep us busy for several years to come, if not decades. So we have a lively, thoughtful, and thriving computational biology community. And it's certainly really nice, like Soren said, to get back the culture and spend time together. So I'd like to thank, first of all, the Simons Institute for hosting us, Ron and Stephen for setting up the month-long program, which this workshop is a part of. Haiyan for graciously stepping up to help with the organization, and Roderick for being such a wonderful co-organizer. We're actually saying on the way back from lunch today that we should think about a way to make this happen. Again, not just stop at this time, but make this more of a recurring event. So the workshop was scientifically productive, I think, but also it was wonderful to be back in person and catch up with people we haven't seen in several years, and people we've known for decades. So last but not least, our heartfelt thanks to all of you, the speakers and the participants, so for your invaluable contributions to the workshop, and keep in touch, safe travel, and hope to see you soon. Thank you all.
Okay, so thanks, Roderick and Sandrine. It's really great to see this actually happening after all this long wait. Really, it was superb, scientifically and otherwise. I thank the Correct Foundation for funding this activity, and the Simons for the organization and the hospitality. For those who are staying around for the long program, you may have already seen the email. We continue the activities. There are 3 meetings next week. On Tuesday, a reading group where the student will present. On Thursday, Martin Vingron, who is joining the long-term visitors, will give a talk. And on Friday, we have a talk from an Illumina scientist, and we have plans also for the following week, which will be the last. So thanks again for coming, and for those who are leaving, have a safe trip home.
I just want to echo Ron's comments and thank all of you for your participation. Meanwhile, the Core at Berkeley Tel Aviv initiative continues beyond this for another about 1.5 years. We'll have another round of grants, which will be open this fall, free to apply for. Those of you from Berkeley and Tel Aviv, we encourage you to participate. And we'll have another set of workshops joint between the campuses going on there. And so to Sandrine's thought, how do we continue this? That's something that we definitely hope to continue that conversation. And glad to hear that you're enthusiastic to do even more work for us after all these years of putting this together. So thank you all very much, and I look forward to seeing you later today and for as long as you stay here. And happy travels.